Hello friends, today I'm going to give you a brief overview about the learning module that we have developed with the help of NIH and Google. So this is the module that we have developed to show the integration of genomic information together with the epigenomics. Most of you must be familiar with the genomics and genes, DNA sequences, but if we go beyond the genomics, then we talk about the epigenomics. Epigenomics is the study of how the cell controls the gene activity without changing the DNA sequence. As epigenomic changes help to determine whether the gene is turned on or off, they influence the production of protein in the cell. There are two kinds of process that we study in this section is histone modification or DNA methylation. So here we are just going to talk about the DNA methylation. But it is very important to understand the genomics together with the epigenomics. The scientific knowledge about the epigenomic regulation together with the genomics is not very well studied. It is crucial and the fundamental concept to study the combined effect of genomics and epigenomics, and it should be taught together for the coming generation in a very simple and precise manner so that we can improve the research outcome. Therefore, we designed this module to study both the components, that is genetics and epigenetics, in terms of DNA methylation at the same time, and we can explain changes in terms of the expression of genes together with the controlling factor of the gene regulation in different phenotypic conditions using the next generation sequencing data. If we talk about the history, when the first human genome was sequenced in 2000, so people were expecting that they will decode all the possible reason of all possible disease related to the human health. But after a short term, they realized that there is something still missing, which is the controlling part of the genome. And in 2010, it has made a cover page of Times magazine, where they first describe about the epigenomics, and they mention that this is something which is beyond the genetics and which controls the genetic component. So here in this module, we have covered all the possible process that we use to combine transcriptomic and epigenomics together. As we start with the raw data, which comes in a FASTQ format. And as any other FASTQ format, we first go through the quality control report of FASTQ file. And if it requires, we use streaming and other quality control process, and then map it with the reference genome, then count the features or methylation extractions. Then we normalize the data before we perform the differential methylation or differential expression analysis. Since there is no direct relation between DNA methylation and gene expression, we'll go through some approaches such as to perform a correlation between DNA methylation and expression data, to find the overlaps and engagements, to find the common functions and pathway related to the both of the differentially methylated or expressed gene. And at the end, we'll try to find some small segment in terms of motif search. So for this study, we have used a pair data from the same breast cancer patient, for which we have DNA and DNA methylation and gene expression. The first major component for any analysis is to understand the data. And here, if we look at the distribution of DNA methylation data and gene expression data, we will find out that there is a significant difference. If we consider gene expression data is normally distributed data, then when we look at the methylation data, it is bimodal or multimodal distributed data. So in that case, we cannot apply the same test which we are using for the gene expression data on DNA methylation data. And it is recommended to use a non-parametric approach on the methylation data as compared to the parametric approach which we generally apply on the gene expression data. So here we are going to use two of our previous publications that we have used as an example for this module. One is the research outcome of the DNA methylation and gene expression data on a breast cancer patient. The second is more like a method paper, which we have published in OncoTarget, which explain like what all the methods is more suitable on the gene expression as well as the DNA methylation data. Before we go into that, I would like to give you a brief idea about the breast cancer. So breast cancer is considered as a heterogeneous disease, which is mainly classified into four major categories. The least aggressive breast cancer is known as luminal A, and the most aggressive cancer is considered as a basal-like. There are other categories which are based on the proliferation level, such as luminal B, and another category which is HER2 positive. All those categories are can be classified based on gene expression profiles using just one gene or a set of gene. And the outcome of these, all four subtypes are different. The luminal A has a better outcome as compared to the triple negative or basal type of breast cancer patient. So until we published this paper, all the people were using only the gene expression data to make a classification between the breast cancer patient. But in this publication, we have shown if we perform an unsupervised clustering using the DNA methylation data, we found two major clusters. And as you can see clearly, that both of those clusters was a very well separated between a different clinical pathological condition, including the ER and HER2 status. So as you can see, most of the ER positive patient on the one side and ER negative patient on the other side of the other side of the histogram. So this is a kind of reproduction of or the verification of the gene expression data. But can we also go beyond that these findings when we are using the DNA methylation data? So the answer is yes. And then we again classify these samples into 6 major categories. And again, they were very well separated in terms of not only the ER and HER2 status, but also the grade, nodal status, and other clinical pathological factor, which is highly associated with the clinical outcome of the breast cancer patient. The second major component is the activity of gene expression and DNA methylation. And one of the major hypothesis is DNA methylation and gene expression data are anti-correlated. And our interest was to find out is this component or this hypothesis is valid across the entire genome, or there is some specific reason or area of the genome which is supported by this hypothesis. So we classify the entire gene expression data into 3 major categories: low, medium, and high. And accordingly, we found the DNA methylation profile associated with those gene expression profile. And in this figure, you can see that the distance from the transcription start site give the information about the promoter operating region of any genome, which is considered like within the 500 range from the transcription start site. And as we can see, like as we increasing the gene expression data, the DNA methylation level goes down, specifically in the region of the 500 base pair from the transcription start site. So in conclusion, we can say that DNA methylation and gene expression is anti-correlated, but in a specific region, which is from the 500 base pair from the transcription start site. In addition to that, we can also identify like some of the genes which were not significant in terms of the gene expression level. But when we look at the same gene at the methylation level, there is a significant difference in these genes among all the 6 clusters that we identify using the classifier made by the DNA methylation data. So these are the new findings which we find out through the methylation data, but also we can correlate with the gene expression data. Similarly, in addition to that, we found out some of the functions which were associated with DNA methylation and accordingly their relation in terms of expression. And all the other components such as motifs that has been described in this module. I hope you like this video, and we are very thankful that you are interested in this module. Thanks for watching.